Good day, everyone. Good day, ma'am. I am John Alfred C. Churica. So, my topic is plant breeding using biotechnology. So, what is plant breeding using biotechnology? It is a modern biotechnology allows plant breeders to select genes that produce beneficial traits and move them from one organism to others. So, plant biotechnology is a far more precise and selective than crossbreeding and producing desired agronomic traits. Domestication altered modern crop lines from its progenitor. Inflorescences has no cob, allowing the seed to separate and disappear easily when they matured. So compared with modern corn with type that retains seed on the air, leading to develop of the cob. So biotechnology, it is refers to the use of living organism or their components to provide using products. So making wines, beer, or bread, composing materials, releasing parasitics, waste wasp to control insect, pest, breeding plant or animal and producing crops and livestock. So first we have micropropagation. So it is a rapid vegetative propagation of plant under in vitro conditions of high light intensity, controlled temperature, and a defined nutrition medium. So the techniques has been applied to a substantial number of commercial vegetative propagated plant species. So number 2, embryo culture. So it is a technique of cultivation of embryo under aseptic conditions on a nutrients medium. So the method can be divided into two applications. One is performed with mature embryo and help mainly in shortening the period of germination by overcoming seed dormancy. So number 3 is culture. So plant regeneration from the haploid microspore cells with the aim of haploid plant production. So there are few reports on plant regeneration in the culture of buckwheat. So number 4 is cell culture. The removal of cell from an animal or plant and their subsequent growth in a favorable artificial environment. So next we have protoplast fusion. Another method of producing new genetic variation in the laboratory by fusing protoplast. So we have two types. So first is protoplast and the second is under. So first is a plant cell. Protoplast is a plant cell from which the rapid and other cell wall has been removed using enzymes or proteins that break down or degrade the cell wall. So next is under certain conditions, protoplast derived from genetically different plant can be fused much like two soup bundle, allowing their DNA or other cellular contents to be combined. So next, we have number 6, we have marker assisted or molecular assisted breeding. So provide a dramatic improvements in the efficiency with which breeders can select plants with desirable combinations of genes by identifying and observing specific makers. So we have makers. Makers is a generic tag that identifies a particular locations within a plant's DNA sequence. So so we have two types of DNA makers, which is the DNA makers and proteins makers. So DNA makers identifies locations where the sequence differs among varieties or breeding lines. So the protein makers based on difference in the proteins that are synthesized from the genes. So two types of protein makers is easy mes or isozy mes. So easy mes are proteins that catalyze catalyze specific biochemical reactions and can be therefore be assigned for their activities. So isozymes is multiple copies of the same enzyme in the plant that are differ slightly in size or charge, making it possible to separate them from each other. So and the B is polymorphism. Polymorphism is an individuals in a single species which different traits or DNA sequence. Makers can be used as a signpost for the location of this polymorphism. So number seven, we have recombinant DNA techniques. So it is allows the specific identification, isolation, and alternation of genes and their reintroduction into living organisms to produce transgenic varieties. So, supplementing and extending traditional breeding methods. So, in a year 1996 to 2001, global production are for transgenic crops. So, it is soybeans, cotton, corn, and canola. So, it increased from 0 to 50 million hectares. So, Culgin's flavor serve tomato, first commercial food product derived from a transgenic crop variety. Golden rice, accumulation of beta carotene which is converted into vitamin A with consumed by animals in a rice gains. So BT corn that has been inserted with BT toxin gene from Bacillus thuringiensis bacteria in order for the corn to be poisonous to corn border, which alkaline but not to humans with acidic stomach. So, DNA clothing techniques. So, isolating a specific segment of DNA molecules, inserting them into other DNA molecules or vector, and using bacteria as biological copy machines. So, to produce large quantities of these DNA molecules. So, these techniques rely on restriction enzyme that can recognize specific DNA sequence and cut the DNA strands at those sites. So, the end of the DNA are cut. That will be all. Thank you.